Mutations are also caused by compounds that substitute for normal bases, and these are called base analogues, or the compounds that slip between the bases, and these are called intercalating agents, and both of these, they cause errors in replication. So first let us see base analogues. Base analogues are structurally similar to the proper bases but differ in ways. In some ways they are different from the normal bases, and that's why these differences make them treacherous to the cells. Thus base analogues are similar enough to the proper bases to get taken up by the cells and then converted into nucleoside triphosphates by the cells, and finally incorporated into the DNA during replication. This all happen due to some error in identifying these base analogues by the cell. But because of the structural differences from the proper bases, the analogues base pair inaccurately, leading to frequent mistakes during the replication process, because these are not proper bases, so they have some differences. So when they base pair with the opposite strand, here they do not base pair normally, and this leads to mutations. One of the most mutagenic base analog is 5-bromouracil, which is an analog of thymine. The presence of the bromo substrate allows the base to mispair with guanine. Otherwise, this thymine base pair with adenine. But due to the presence of this base analog, it forms the base pair with guanine. Here you can see this is 5-bromouracil, and it is base analog of thymine. So in the normal sequence, here thymine would be present, and in front of thymine there would come adenine in case of normal base. But this is a base analog, and it has replaced A with G, with guanine. So this 5-bromouracil base pairs with guanine instead of adenine. Second such compounds are intercalating agents. Intercalating agents are flat molecules containing several polycyclic rings, and they can bind to the purine or pyrimidine bases of DNA, just as the bases bind or stack with each other in the double stranded DNA molecule. So they also mistakenly they are base pair with the in the DNA molecule. So here are some examples of intercalating agents. This is proflavin, and then acridine dye, which can intercalate into the DNA molecule, and the third one is ethidium. All these are the examples of intercalating agents. Intercalating agents such as proflavin, acridine and ethidium cause the deletion or addition of a base pair or even a few base pairs in the DNA molecule. These intercalating agents also cause short insertions or deletions in the DNA molecule. But how do these agents cause short insertions or deletions? They can cause one base pair insertion, one base insertion or one base deletion. But how they cause short insertions and deletions in the DNA molecule? One possibility in the case of insertion is that by slipping between the bases in the template strand, these intercalating agents, they slip between the bases, and these mutagens cause the DNA polymerase to insert an extra nucleotide sequence opposite the intercalated molecule. So here two strands are present. These intercalating agents get entry into one strand. They slip between the bases in one strand. So when the polymerase machinery will replicate this strand, and when it passes this intercalated agent, it may add some extra bases or extra nucleotides on the opposite strand. So in this way, it leads to insertion. Conversely, in the case of deletions, the distortion of the template caused by the presence of an intercalated molecule might cause the polymerase to skip a single or few nucleotides. Similarly, if there is some intercalation of these agents, the polymerase chain machinery may skip that portion in the template, in the new strand. So in this way, that portion will be deleted in the new strand. So in this way, the base analogues and intercalated agents damage DNA.